Good morning, everyone. For those that don't know me, I'm Lachlan Woodcock and I'm part of SPI's Extension Subcommittee. So today I wanted to give you a quick update on what we've been working on around genetic testing for Tenderness and Speckle Park cattle. It's an area that's been getting quite a lot of interest, and while we don't have all the answers yet, we are making good progress, and I think it's well worthwhile sharing what we have learned so far. Some of you may have noticed that when you fill out the DNA submission form to register your calves, there's an additional test called Tenderness that you can select as one of the add-on tests. When you select that test, your DNA results come back with a tenderness score between 1 and 10 for that animal. We've been doing this in our herd since we first got into Speckle Parks and thought this was interesting and had some relevance. When the subcommittee looked into it, we found there's been over 1,500 animals tested across the breed so far, and that really got us thinking and having a conversation about how relevant this test is to the breed. Can it help us make better breeding decisions, and should we look into it more deeply to understand what it is that we have available to us? Some progress we've already made is that we've got tenderness scores showing up in the ABRI database now. So if an animal's being tested, you can jump online and see the results straight away. But the next step is really trying to understand what those scores are telling us and how we might be able to use them. So today I'm going to give you a bit more background on the test itself, some of the work we've been doing to unpack it, and look at the key knowledge gaps and the next steps forward for the breed. So first, let's unpack what we mean when we talk about meat tenderness. Tenderness is essentially how easy the meat can be chewed or cut. It's one of the qualities that consumers first notice when they eat beef. We all know that tough, chewy beef leads to a disappointing eating experience, and ultimately, our goal is to produce exceptional beef. Tenderness is measured through a post-slaughter evaluation known as the Warner-Bratzler shear force test. In this process, a piece of meat is extracted from the carcass. This is cooked, cooled, and cut into small cores that are then sliced with a V-shaped blade, and a measurement of how much shear force is required to slice through the meat is recorded. The less shear force or the lower shear force, the more tender the meat is deemed to be. The results are usually recorded in pounds or kilos, depending on the data set. Those shear force values correlate highly with findings from consumer panels, so it's a really reliable method of measuring tenderness. However, the obvious challenge is that it is a destructive test and the animal must be slaughtered. So what influences tenderness? Tenderness is influenced by a range of factors, including breed, the age of the animal, nutrition, handling of the animal, and post-slaughter processing. So we know things like younger cattle are going to be more tender than older, more mature cattle. We know that if carcasses are hung via the tender stretch method, that they're going to be more tender than if they're hung under normal methods. We know that if animals are stressed before slaughter, they're going to be less tender. We also know that genetics play a significant role and that tenderness has a moderate heritability, which means that we can select through it. And this is where our genetic testing becomes valuable, because if we can identify markers associated with tenderness, we can predict tenderness in live animals, allowing us to make decisions before they hit the abattoir. It's really important to highlight that tenderness is a polygenic trait, which means it's controlled by multiple genes rather than just a few. However, early research into genetic control focused on key genes that regulate enzymes in the calpain and calpacitin system, which are responsible for the breaking down of the muscle proteins after slaughter. And these are the genes that our genetic testing was based on. So to understand the genetic test that is used, we also need to understand the system and how it influences tenderness after slaughter. When an animal is slaughtered, its muscle stops receiving oxygen. The muscle fibers tighten and it goes into rigor mortis. Over time, the meat becomes tender again, and that's largely thanks to this system. Calpains are natural enzymes in the muscle which are activated by the calcium that is released from the muscle cells post-slaughter. They start gently breaking down some of the structural meat proteins. It doesn't destroy them, but it just loosens the connections between them, and that's what makes the meat more tender. Animals that have high calpain activity tend to have more protein breakdown, and as a result are more tender. But in all things biological, there's always an inhibitor called calpastatin, and its job is to block that process. Animals with higher calpastatin activity have less protein breakdown, which generally means tougher meat. So it's the balancing act between these two tenderizing effect of the calpain and the blocking effect of the calpastatin that determines how much muscle protein is broken down during the initial aging process and how tender the beef is. We know that different breeds and animals within breeds can have different natural levels of these enzymes, and that it is driven by genetics, and that's why the calpain and calpastatin genes were at the foundation of the initial tenderness tests that were developed. Back in the early 2000s, researchers started to explore whether we could find genetic mutations linked to tenderness in beef cattle. The way they did this is they built reference populations from which they collected shear force data and then looked for genetic markers that significantly correlate with those measurements. From that work came two main commercial tests. The first is the GeneStar tenderness test, and the second is the Neogen Ingenuity Tender Gene test. The GeneStar test was released in the early 2000s and evaluates 4 markers in 3 genes, 2 in the calpain and 2 different calpastatin genes. It uses a star system, so you get up to 2 stars per markers, 1 to 8 stars total, and 8 stars in the most is the most tender. The test we have access to at SPI is Neogen's Ingenuity test, and that one looks at the same 2 markers in the calpain gene but has a different calpastatin gene. It produces a score from 1 to 10, with 10 being the most tender. The Ingenuity test looks at one calpastatin gene, which is called CAST, or specifically UOG CAST1. And at that site, there's either a C or a G. Animals with a CC have decreased enzyme activity. The decreased activity means decreased inhibition of calpain, which means higher tenderness. The other 2 components that are used are 2 markers in the calpain gene, or the CAPN1. It looks at a location of CAPN316 and CAPN4751. There is options for C or G at 316 and C or T at 4751. And if there are 2 Cs at those sites, we get increasing activity of calpain and more muscle breakdown. The test then combines those 3 sites together to give you a tender score from 1 to 10. So, for example, if you have a CC at all 3 sites, you get a tenderness score of 10. If you have a GG at 2 of them and a TT at the other, you get a score of 1. This table shows how the SNPs at those markers combine to form a genotype, which infers the tenderness score and the associated shear force value. The data set came from the initial 1,209 animals that were part of the reference group for marker development. This reference population was made up of Brangus, Charolais Cross, Angus, Red Angus, and Brahman cattle. It is likely that the shear force values associated with the genotypes will slightly change depending on the experiment or under the conditions in which they're undertaken. But overall, there's a clear pattern. The greater the number of beneficial alleles, the more negative the shear force. It is important to recognise that Speckle Parks were not part of the validation population for these stats, so we need to keep that in mind when we interpret the data. But speckles are genetically close to Angus, so we can have some confidence. What you'll notice straight away from the table is that there are 27 potential genotypes and only 10 scores. Scores 1 to 7 have different genotypes associated with them. So just getting a score from an animal doesn't really tell us a lot to be able to make breeding decisions about what we would mate together. We've recently found out as a committee that these markers are part of the 50 and 100k SNP that is offered. And so we can actually now access them. All new registered animals from now on will have their tenderness test completed as part of the test bundle and receive the genotype of their animal as well as the score. So how do we use this data? That's kind of the next question. I mean, if an animal is tenderness 1, it doesn't mean they go on the coal list straight away. It just means you might need to be strategic on what you mate it with. You can go from a score 1 to a score 10 in 2 generations if you get the mating correct. It just gives us another piece of information or data that we can use to make sound breeding decisions about what we may mate together. I mentioned before that breed has an impact on tenderness, and some studies have examined the frequency of the different alleles for each of the different markers. The Ingenuity tests have been used on a range of different cattle breeds with various levels of significant gains identified from each of the markers, particularly when you look at temperate versus tropical populations. It's really interesting data when you start to look into it. In this table here, I've just pulled out 3 breeds. We've got Red Angus, Brahman, and Black Angus. And what you'll notice is that the Brahmans actually have a really high proportion of TTs and GGs for those first 2 markers, and more of the unfavourable alleles for the third. We know that Brahman cattle produce tougher meat, and so you can see why when we cross Brahman cattle with other breeds of cattle, we potentially get a more tender product. This may have implications if we are selling cattle into the northern markets. The other thing we need to be aware of is that these enzymes naturally occur the way they do because they've been naturally selected that way. There is some discussion that tropical cattle have low cow pain activity and high calpastin activity to allow the animal to have low metabolic heat production. By having lower protein breakdown, which means they can then handle the hotter conditions, which may or may not be important for those breeders selling into northern markets, and something that we need to take into consideration as we learn more about this test and what it means for tenderness. Other breeds have looked at using this data as a marketing tool by looking at the distribution of the tenderness scores. The Red Angus Society say they have a higher average tenderness score than the reference population, and the Belted Galloways did a similar thing. But we really have to remember that tenderness is a polygenic trait. When they rolled this test out into larger cohorts of animals, they started to find that the tenderness scores didn't really explain all the variations in tenderness or shear force that they were seeing. It was probably oversold a little bit in what it could actually do. And that's due to 2 factors. A, it's due to the fact that the reference population was a reference population, so expect the marker to be highly correlated within that population. And B, the selected markers and genes contribute largely, but meat tenderness is influenced by many other traits and factors. And so just as a really quick example of that, say you have 2 animals A and B, both tenderness score of 10, but one has a docility score of 10 being super docile and quiet, and one has a docility score of 2. That animal gets stirred up really easily in the yards. When those animals are slaughtered, it's highly likely that animal A would have more tender meat because animal B is going to get stressed out at slaughter. We know animals that are stressed at slaughter have higher meat pH, and we know that a higher meat pH decreases cow pain and increases calpastin. And that's just a simple example of how 2 traits can come together to give you an overall phenotype. We also have to keep in mind in the last 20 years, we've had a significant increase in our ability to generate and analyse genetic data. We are now doing 100 case SNP and doing single-step genomics. At some point, hopefully quickly, a more informative genetic test will catch up, but it's also going to be breed-specific, and we're going to have to be the ones to drive that. And Angus Australia have already developed a research breeding value for shear force. Angus Australia have been working with the Animal Genetic Breeding Unit, UNE, and the researchers at ABRI to develop and release research breeding values, or RBVs, for shear force. This has been done as part of the Trans-Tasman Angus Evaluation Program. The shear force RBVs are calculated from laboratory shear force measurements, pedigree, and a little bit of genomics. Lower, more favourable values are better, but these are just based on the shear force phenotypes. So they've looked at which animals have more negative shear force, which animals have sired those, and used all that data with some fancy stats to generate these RBVs. They have not yet done any genetic correlation work because they don't think they have enough numbers yet. But my assumption is that they will eventually take all the SNP data that they've got, and again we know that the cow pain and calpastatin genes are part of that data set, to identify SNPs or QTLs that explain the variations observed. All right, so to wrap up, just a bit of a summary on our key takeaways. So the point of the presentation was to try and make sure that everyone has enough background information on this test, so you guys can go away, do some of your own reading, and learn a bit more about this. So to quickly recap on the key messages, Tenderness is an important part of meat quality, and it can only be measured after slaughter using the shear force test. The phenotype of tenderness is a combination of environment, management, and genetic factors. Tenderness is a polygenic trait and is controlled by many genes, but SNPs in the calpain and calpastidin genes are responsible for some of the variation we see, but are not 100% causal. The significance of the impact of these markers is breed-specific, and Speckle Parks were not included in the identification of the validation of these markers. The Neogen Ingenuity Tender Gene Test provides a tenderness score from 1 to 10 and will now provide also the genotype. All new registered animals will have tenderness added to the genetic bundle at no extra cost. Tenderness animals are identifiable on the database by tenderness score and soon by genotype also. There is a potential opportunity for the Speckle Park breed to develop an RBV for shear force out of this information that we are collecting. So what do we do now? We're trying to generate data. We want to be able to actually identify the genotype of as many animals as possible to give us an indication of what background genetics we've already got within the Speckle Park breed. So you would have noticed there's been an email sent around for people to put in expressions of interest to have their animals genotype for those tenderness scores. The key questions we will still need to answer. Does the genetic test correlate to meat tenderness or shear force in Speckle Park? How does the number of Speckle Park genotypes compare to other breeds? Are there any other SNPs that would be relevant for us to look at? How do we validate the accuracy of the genetic data? Can we test shear force for a cohort of animals? If producers aren't paid for tenderness, does this actually matter? So, that's a wrap for my presentation on tenderness. I hope that was informative on where we're currently at with the subcommittee on working on tenderness. I'm excited about the next steps. Please reach out if you've got any questions, and please submit animals for the genotype data via the JOT form to SPI. This is critical to the validation work going forward. Thank you for the opportunity, and that's me.